to introduce Anass Bouchnita, our today's speaker. Anass works at the University of Texas in Austin now in the group. Maybe you might say, if you want, then a couple of words about your group and people around, and it's up to you. As far as I understand, it's a group working on modeling of COVID-19, and the title of our today's presentation is a new class of COVID-19 compartmental models that explicitly incorporate the changes in population immunity. Please, Anass, you are very welcome, so you can start your presentation.
Thank you very much, Vitaly, for this presentation and for the introduction, and also for the invitation to give this seminar. So yes, I work at the UT COVID-19 Modeling Consortium, and it's a local group at the University of Texas at Austin that aims to use mathematical models to detect, combat, and mitigate COVID-19. So these are the collaborators with whom I've been working, and we also collaborate with public health officials at the city of Austin, at the state of Texas, and in the US, and also with the World Health Organization, as I'm going to show. So this would be the overview of my presentation. I will start with an introduction or a background of why we developed this new class of mathematical models, and I will speak about their derivation from numerical simulations of a more complex within and between host model. And I will give two applications. The first one concerns the impact of SARS-CoV-2 booster dose on COVID mortality, or the optimization of the timing of introducing booster dose in different countries. And the second is the impact of Omicron emergence in the United States. As you know, the landscape of COVID-19 is becoming extremely complex in the last few months. We have lots of new effects that influence the transmission of and severity of this disease. So we have the continuous emergence of new variants of concern that can change the transmissibility, the severity, and the immune escape ability of this virus, and we also have competition between strains sometimes. We have also the vaccines, and here we have questions related to vaccine efficacy, as we have different type of vaccines. And we have the immunity-related processes. So these are processes related to population immunity, like the waning of immunity, the diversity of sources of immunity. So we have immunity from natural infection versus vaccine-induced immunity, and also how well each immunity works against each type of variant or each type of infection. And more importantly, there is an interplay between all of that. So we know that immunity reduces the transmissibility and severity of the variants of concern, and so does vaccination. On the other hand, the variants of concern reduce or escape to the efficacy of the vaccination, and can result into diversity in the population immunity. So we can have, for example, immunity from Delta, immunity from Omicron, and so on. And also we know that vaccination increases the immunity. And so what's happened is that lately the mathematical models that we are using to describe COVID transmission dynamics need to incorporate all of these effects, and as a result they are becoming more and more complex. And so if we speak about what changed in the modeling of COVID-19, is that we moved from oversimplification at the very beginning of the pandemic, because at that time most models did not include all important effects like asymptomatic and pre-symptomatic transmission, reinfections, and so on. But now we have a lot of data, we have a lot of knowledge, and we have all this interplay and this complexity, and this makes models very complex. So if we take a look at what researchers are using now, we find two main types of models. We have the compartmental models that are getting stratified according to immunity levels, for example, or vaccination status, and sometimes also to strain status or variant status. So we can have a group of populations stratified according to immunity, variants, vaccination, and so on. And on the other hand, we have the individual-based models, and these are like the agent-based models that explicitly track what's happening inside each individual. And recently, these models also try to implement within host compartments, or at least try to track the level of antibodies in each individual and how this translates into protection against infection. And as you can see, this has resulted in very complex models that can be difficult to interpret and also to implement. And sometimes we need to introduce some additional assumptions. And here the most important example is how to handle booster doses. So if we want to handle a booster dose using such models, then we need to make assumptions regarding who is getting these boosters first. And we don't know if this is going to affect the obtained results or no. So these are some of the problems that we are currently facing as a community of modelers, and that we are trying to overcome using some new methods. And one of them is what I'm going to present today, which is a new class of epidemiological models that explicitly describe the changes in population immunity and how it affects the severity and the susceptibility of COVID-19. And as I said, I will give two applications where we have used these models. But first of all, I will discuss the derivation of the model. And to understand this, we were working on understanding the effects of population immunity on the epidemiological characteristics of COVID-19. And we have started developing this type of modeling framework where we have an agent-based model, where each agent can move according to some model that I will describe later on, what's called a social force model. And inside of each individual, there is an independent within host model that describes the viral kinetics and the interaction between the infection and the innate and adaptive immune responses, as you can see here in that figure. And so this model couples the within host viral kinetics. So these are what's happening inside of each individual in terms of interaction between the virus and the immune response, and a social force framework. So this is the agent-based framework that we are using for infection transmission. And we assume that the evolution of the disease depends on the area under the curve of the viral load. So the more viral load is generated inside of the individual, the more are the chances that this individual will develop symptoms, get hospitalized, or even die. But this depends on different variables like the age and the comorbidities that this individual has. And also we assume that the chances of transmitting the virus depends on the viral load in the infectious person. So this is the within host model that we are using, and as you can see, I think in your group here you are probably using something very similar. But basically the idea is that we have this cell limited model that we extend by adding equations for the adaptive immune response. And we want to do this because we want also to keep track of the evolution of T cells, B cells, and antibodies after the resolution of the infection. So we have some healthy cells here that can get infected, and when they get infected, they secrete the virus, which can also, of course, contaminate other cells. And this can be stopped by this interferon alpha, but at the same time we have this activation of B cells and T cells that can either eliminate the infected cells directly or secrete antibodies that eliminate the virus. So this is the model that we are using, and we are assuming that there is a delay for this part of the adaptive immune response. Another important point is that some of the parameters are individualized to account for the effect of age and comorbidities. And this is what I'm going to explain in the next slide. So, for example, the age determines the capacity of naive lymphocytes. And this is what a study has shown, is that younger population tend to have a higher reservoir of B cells and T cells, and so they can generate a more robust adaptive immune response. And also we need to account for the half-life time of the memory immunity. So this is the number of antigen-specific T cells and B cells and antibodies after the infection, and we have taken this from an experimental study. And for the remaining parameters, we are using some available estimates from the literature, but we are doing a very thorough validation, first of all by comparing by fitting to viral loads from an experimental study. And also what I'm going to show, not on this slide, but on the next one maybe, we are also doing a virtual clinical trial, like a clinical trial with a cohort of virtual patients. But before moving to that, I want to say that the evolution of the disease is modeled through this area under the curve of the viral load. So this is like the integral of the viral load, and the evolution of this estimate determines whether this patient will have symptoms, will get hospitalized or die, as you can see in this curve. And these thresholds for hospitalization and death can depend on the age and on the comorbidities of the individual. So this means, like, for some individual, they can support like high or they can support being exposed to higher viral load than others, depending on their health, whether they have some comorbidities like diabetes or some cardiovascular or respiratory disease, and also depending on their age. So as I said, we have to make a validation, and we had to make sure that the model can be incorporated into an agent-based model and give us some good epidemiological features.
Anas, sorry to interrupt you. Do you mind if we ask questions during your? Yeah, then Alexei, please. Alexei Tokarev has a question. Go ahead.
Yeah, Anas, could you please return to the previous slide? I have a question about area. No, next one. with yes. So you deal with area under the curve, and if it is above some threshold, then man is hospitalized. But the question is as follows: as far as I understand, the more serious symptoms are somewhere on the peak of the curve. So when we reach the peak, there is the highest, yes, somewhere here, the highest probability of to be hospitalized or not. And if it is not needed, then the person stays at home, and it is not needed, and he stays there. So could you please tell something about?
Yeah, thank you for this question. I think that's a very important point, but I think it really depends on the literature, because in some other works they show that in this day before the peak, then it's probably the symptom onset. So this is where the symptom starts, just like one day before the peak. So maybe somewhere here, and this is what we have used here. And also we have also, as I'm going to show next, we have made sure that the different characteristics of the evolution of the clinical evolution of COVID mimics the values that are clinically observed. I think I have also come across some works suggesting that in the peak people can get hospitalized on maybe 1 or 2 days after the peak, but it can really depend on the patient. What I'm going to show is that, for example, in this table, all of the different periods were fitted to be within the range of what is clinically observed. So as I said, we wanted to make a validation for this clinical and epidemiological characteristics of the evolution of the disease, and we have generated a cohort of 1,500 virtual patients, and each of these patients will have a specific age and a specific risk group. So we have considered 3 risk groups: patients with comorbidities, and normal patients, and patients with a strong immune response. And also we have considered an age tree that mimics or corresponds to the distribution of ages in the US. And as I said previously, the area under the curve thresholds for hospitalizations and symptomatic disease and so on were calibrated to approximate the epidemiological features of COVID-19, and this is a comparison between the model outputs and the actual values in the literature. So, for example, the model predicted that there will be about 70% of symptomatic cases, and 14% of them will be hospitalized, and also the case fatality ratio is within the observed values. The time of tested positive is very important because we couldn't find this in the literature, but there were some recommendations from the Center for Disease Control suggesting that the PCR tests are not accurate during the first 3 days. And our model suggests that it takes about 2.3 days to test positive after an infection. An important caveat of the model, or a limitation, is that the incubation period does not change too much. And I tried fixing this, but when I changed the characteristics of the viral load too much, then I cannot find a good estimate for the other parameters. So I think the work that you are doing for the incubation period is very important, because you are changing this initial viral load and seeing how this impacts the characteristics. But I tried doing this too, but when I change the initial viral load, then sometimes I find some physiologically irrelevant cases where, for example, the infection does not resolve and problems like that. So the easiest problem that I found is to take like the same viral load for each infection, is to fix the initial viral load. But of course, this is a limitation that I think we should try to overcome in the future. Also, the time from hospitalization from symptom to hospitalization, and the length of stay, and also the time from symptom onset to death were within the range, although there are different values in the literature. So the next step would be to take this model that we calibrated to replicate both the experimentally observed viral load and the clinical features of COVID-19, and embed it into an agent-based model. And for that, we have used this framework called a social force model, in which each individual is like a point that is subject to Newton's second law of dynamic, and namely that there are three different forces: the social force, the self-driven force, which describe the tendency of the individual to move toward a certain direction or to reach a certain target, and a repulsive force from the wall. And as I said, the social force describes the tendency of individuals to maintain a social distance from other person. And to approximate larger systems, we are using periodic boundary conditions. So we need to find a way to describe the cis transmission, and one of the simplest way to do it is to assume that it depends on the viral load of the infectious individual. And so when a person has a high viral load, then there are higher chance that this person will transmit the virus to a susceptible individual. And so this is the probability that we are assuming for the chance of transmission during contact. So when there is a contact between an infectious individual and a susceptible one, then we evaluate this probability, and if it's good, then we assume that the virus is transmitted. But we have this parameter here, this positive constant, and it can really modulate the spread of the virus. So if alpha is big, then of course the probability will be low, and this means that the virus will not be spreading too much. So we had to fit this parameter such that we have a basic reproduction number between 2 and 3, and this was the basic reproduction number for the COVID-19 that was first appeared, or the wild type Wuhan strain. So this is what simulation looks like, and you can see that different people are getting infected and becoming symptomatic or getting hospitalized. So we consider 500 individuals, and 3 of them to be exposed to the virus initially. And as you can see that even though we have considered 7 months of the simulation, then there is always some level of people who are getting infected, and this is because there are these chances of reinfections, and this is more approximate to what is happening with COVID than using SEI-RS model, for example. We also did some validation.
Sorry, Anass, if you can return to the previous slide, because you mentioned, for example, a reinfection here. So what are the exact conditions here? So there are many things you are presented, but for example, in these simulations, you have reinfection, means people become like recovered, become again susceptible, right?
Yeah, yeah.
After some time, yeah.
Yeah. That's very, yeah.
And do you have vaccination in this model and in these simulations or not?
No, for this model we don't have vaccination, but just so I can go back to your question regarding reinfection. So what happens in reality is that there are always possibility for reinfection, even though if someone was just infected before. But what happens is that this person will have a high level of antibodies such that they will not test positive, because even if they get infected, if they have like a high level of antibodies and antigen specific T cells, then they will not test positive. But after some time, then the number of antibodies will start dropping, and also the antigen specific T cells. And if they get infected, then they might test positive, but the chances that they will be hospitalized will be or develop symptoms will be low, or depending on the antibodies that they had.
Yeah, no, I understand that. Yeah, I agree completely with you what to say. But my question concerns exactly the model which you use here. So you have here, so recovered, I mean, here in this modeling, recovered become again susceptible after some time, no vaccination. The last question to this particular modeling, is the motion random here?
Yeah, good question. Yes, for this particular model, we are assuming that this self-driven force to be, so we have this desired velocity, and we are assuming that the desired velocity is points toward random directions, but with the pedestrian speed.
Okay. Thanks. Continue, please.
Yeah, thank you for the questions. So we also did a validation regarding the serial interval time, which is, I think, the difference between a person getting showing symptoms or an infector showing symptoms and an infectee showing symptoms. So it's like if someone infects another person and shows symptoms, and this other person shows symptoms, then what is the duration between this? And we found values that match the that not only match the what's in the literature, but also the distribution of this value can be approximated by a log normal distribution, which is also in a good agreement with the literature. And also in terms of pre-symptomatic infections, then we found that about 35% of infections occur during the pre-symptomatic period, which seemed to agree with a previous study. So now we try to see what happens in terms of population immunity, and to do this, we try to model or to visualize the level of antibodies in the individuals as the simulation evolve. And as you can see, at the beginning, most of them, they don't have any antibodies, or after 1 month. And after 2 months and 3 months, then there are few people who have high level of antibodies, but after sometimes there is like a diffusion effect, or like this, as you can see here in these histograms, there is this levels of antibodies going to becoming a little bit close to zero as the time progress. And so we finish with something like this, with a more diverse landscape in terms of antibodies. So at the beginning, all of the individuals either have 0 or 1. So few of them have a high level of antibodies, but as the time progressed, then there is some sort of homogenization where most of them, they have some level of protection. And so this is what happens in terms of the effect of population immunity on the model dynamics. So we tried to look at both the average or the median of the antibody titers in the population and compare it with what's happening in terms of population dynamics of symptomatic cases and number of hospitalizations and number of or the infection fatality ratio and so on. And so it seems that having a high level of natural infection immunity does not lead to the complete eradication of the virus, because as I said, there will be always some people getting infected, maybe not going to hospital, but still able to allow the virus to replicate and transmit the virus. But it seems that having a high level of immunity in the population reduces the rates of infection, symptomatic disease, hospitalization, and death. So if you compare this graph with this, for example, this graphs here, then you can see that in this period where the levels of natural immunity is high, then the numbers go down. And after it starts waning or it starts decaying, then there seems to be a new wave or like the beginning of an increase in cases. So we tried to analyze these results and come up with a model that is low dimensional and very simple, that can incorporate all of that. And so, as you know, most of the existing compartmental models, they assume that there are two groups of individuals. So individuals can be either fully susceptible or they are recovered or removed and cannot contract the virus. But it seems that according to the insights that we got from this model and what we know about COVID-19 in general, it seems that this susceptibility to infection is relative. And this is because we have evidence suggesting that vaccinated individuals have low chance of getting infected, but there are still some breakthrough cases, some cases of people who get infected although they are vaccinated. And also the same goes for natural immunity, but here we are talking about reinfections. So people who got infected once getting infected a second time. On the other hand, we know that there are some people who were in close contact with infectious individuals but did not contract the virus. And in general, the results of the multiscale model suggest that population immunity reduces the rates of susceptibility and severity of COVID-19 in general. So we tried to introduce this into a simple model, like the simplest model that we can imagine. So the idea is that we introduce a specific state variable for immunity called M, that you can see here in this diagram here. And we describe the change in this state variable using a specific ordinary differential equation or a specific equation. And we assume that the value of this M regulates the susceptibility, the chances of developing symptoms, the hospitalization rates, and the mortality rates. And so the obtained model is like this. So if you take this equation and if you look at the terms here highlighted in blue, then you see that, for example, this term of susceptibility depends on the level of immunity in the population. And we have this K1 that can be obtained through efficacy of the protection. So we can, for example, assume that M is equal to 1 we know that, for example, if all the population is immunized, then there are still, for example, 20% of people that get infected. And so we can determine these parameters K1. However, there is a parameter, so this is very important if we take a look at the equation for M. And so here we have added a term for vaccination because the model allows it, and it's very simple to introduce it. But also for this term where we have natural infection, we have this term of saturation. So there is a saturation term here where we divide by this 1 plus K4M. And the reason for that is that we observe that when people have already some level of antibodies, then when they get infected once again, they do not generate a lot of antibodies because the magnitude of infection is very small, or is relatively small. And so when they generate additional antibodies, then the chances that they generate low level of antibodies, and so they contribute only only a little bit to the population immunity. And so we fitted the model, and as I said, we had some unknown parameters, but these parameters K1, K2, K3, they we can a little bit adjust them to match the protection levels. And for if it's immunity that comes from vaccination, then we are talking about efficacy of vaccination in blocking infection and hospitalization and so on. But this term K4 need to be fitted, and when we do so, and also of course we need to fit the transmission rate and maybe some one or two other parameters, then we can, as you can see, mimic the results of the multiscale model. So this simple model can really describe the dynamics observed in the numerical simulations with the multiscale model. So we see that the number of symptomatic cases decreases but does not go to zero. And we see that the cumulative death increase but tend to converge to a plateau or stop increasing too much. And also, for example, the rate of symptomatic to total infections also start to decrease and stabilize. And if we look at this immunity state variable M, then we can compare it to the level of the antibody titers in the population. So it seems to increase when the level of antibodies in the population in this fine-grained multiscale model increase, and also it's decrease with this model. So some of the results that we obtained using very simple simulation of this model is, first of all, as long as we have immune waning, even slightest level of immune waning, then it's impossible to obtain a disease-free equilibrium. So there will be always some circulation of the virus in the system, and even if we take like a very small value of immune waning, as you can see here in the figure, then there will be always some level of the virus after this first wave. The second most important result is that booster shots would be required to control and keep the number of hospitalizations and death under a certain level. So if we don't have booster doses and only organize one campaign for vaccination, then what happens is that when this campaign stops, and here we have run very long simulation, like 25 years, but with the same transmission rate. So if we added seasonality, then we might see different waves. But at this point, we were interested by the endemic dynamics of COVID, so we didn't consider that. And so as soon as we stop the vaccination, then the the curve goes back to the stable solution. And also in terms of the level of immunization, so what happens for immunizations is that when we have this vaccination, then we increase immunity, but because of the waning, then these levels go back to the normal level of immunity, and so also the number of daily symptomatic, daily cases or daily infections also goes back to normal. But when we added booster dose, then of course we can keep these numbers at a certain level, but we know that probably it's not possible to have these booster doses all the time. And yeah, I think we see that for the hesitancy level with the third shots, they are much higher than the primary vaccination hesitancy levels. But if we want to keep this number of hospitalization below a certain level, then we need to have booster doses. Now, the good news is that, of course, now we also have this a lot of treatments that are being tested and in clinical trials, so they might also help keeping the number of hospitalizations and death low. So another result that is also interesting is what happens when new variants of concern emerge. And here we only evaluated the inherent transmissibility. So we tried to see what happens when we increase the basic reproduction number. So this is what the variants of concern do, although recently we also have immune escape and changes in the severity. And what happens is that when we increase this basic reproduction number, for example, by like 2-folds, then even if we have a high level of immunity, then the basic reproduction number will be always equal, always superior to 1, which means that it will be impossible to reach herd immunity, because we know that reaching herd immunity means keeping the basic reproduction number inferior to 1. So this means that vaccination will not be sufficient to control some highly transmissible variants of concern, and that probably we need to combine it with face masks and other non-pharmaceutical interventions. Yes. So yeah. So this title is wrong, but yes. After having this low-dimensional model that seemed to be given like very nice dynamics of both the pandemic and endemic stage of COVID-19, we had this request from the World Health Organization SAGE. It's the working group of experts on immunization that give consulting to World Health Organization to see what they do with everything related to vaccination, especially for the middle and low income countries. And they had this problem that they wanted to share with different groups working on modeling COVID-19. And so the problem was that at what overall population vaccination coverage it becomes optimal to start giving booster dose to high-risk individuals instead of giving primary series to lower risk groups. So that was in September, like after months of the vaccination campaign in different countries. And some countries wanted to introduce the booster shots, but they didn't know at which timing they should do it, and what could be the effect on, or what could be the best timing that can reduce the hospitalization and death. And so we wanted to answer this question, and we were discussing, of course, there are many factors, like because they asked us to consider or to perform a multi-country scenario analysis, which means to consider different countries from different background, like countries from middle or low income countries, and also high income countries, and from different regions. So we were discussing, and it seems that there are different factors like vaccination history, contact patterns, population demographics that might affect this decision. So we wanted to understand what is the impact of all of that, which means the contact pattern, the vaccination history, the type of vaccines administered, and so on. And also we wanted to consider the assumptions that were given to us by this group that made this request. And so they wanted us to consider different scenarios related to immune waning, because up until now there are different studies showing different speeds of immune waning depending on the type of vaccines and also depending on the population and so on. And so we had three scenarios. The first one is slow immune waning. So that's scenario one, where immunity does not wane against hospitalization and mortality, but it does wane against infection. And scenario two with moderate immune waning. So the speed of immune waning is 2 years for hospitalization and 6 months for infection. And scenario three with fast immune waning, and here we have very small half-life time for the waning of immunity. So when we had this problem at the beginning, we were still developing this model that I just showed you, and we wanted to make this study using like traditional approaches or like the type of models that other groups are using. And so we devised this type of model where we have an SEIRS model that you can see here in this scheme, but it's stratified according to different levels of protection against hospitalization and protection against infection. And so in total we had 9 groups. So it was like considering this, but 9 times of these compartments that you are seeing here. And after infection, the recovered individuals goes to this compartment. So it's the compartment with the high level of protection against infection and death and hospitalization. But after some time, there is this effect of waning. So these individuals might go to different levels according to the different speeds of immune waning. And we have vaccination that take these individuals and put them into high levels of immunity or of immunization. So what's happened is that when we obtained some results, but we couldn't derive any conclusion. So we had some results, but we had some differences between the countries, but we couldn't tell what's causing this. And this is very normal because the model is very complex, and so it's very hard to tell which group is making the major contribution and so on. And the second problem is that we had to make some additional assumption regarding the allocation of booster doses. It means that when we have the record of people who got the primary vaccines, then after sometimes they propagate along this network here. And so if you want to allocate the booster dose, then there are different ways to do it. Either you have to allocate it in a uniform manner, like across all the groups, or you have to make some sorts of prioritization. And when we make these additional assumptions, then we also obtain some different results. And so this is the type of problems that we are facing now with the traditional modeling approaches, and that makes the interpretation of the results very hard. And so we decided to take this model that we developed explicitly introduce the immunity in the population, and to change it or to adapt it for this specific problem. So here, instead of considering only one state variable for immunity, we are considering two state variables. One that block the infection and symptomatic disease, and the second that block hospitalization and death. And so the reason for that is that we have two speeds of immune waning according to the scenario that I showed you for each population immunity. And also this model is only stratified according to age. So we have 6 age group because we want to introduce vaccination across each age group. And each age group also has different hospitalization rates and symptomatic rates and so on. And as you can see here, it's very simple to introduce vaccination because both the primary vaccination and the booster dose can be introduced in a similar way, only through the number of allocated doses. And so what we consider is that we have this time-dependent function that gives us the number of administered dose for each day, but we consider that it only takes effects on the population immunity 2 weeks later to account for the delay between vaccination and its effects. So this is what the model looks like in terms of equations. And you can see here that we have these two equations for population immunity. So this term is for the upregulation through infection. This one is upregulation through vaccination. I forgot here ML and here MLH. Here we have this, for example, here we have the inhibition of this immunity infection, and here we have the inhibition of this immunity against hospitalization and death. Yes, so we have considered 6 countries with different demographic and contact data. So these countries are Brazil, France, Peru, South Africa, Korea, South Korea, and United States. And we have considered their different contact patterns. So as you know, there are different chances of mixing in different populations across the ages. And also we have considered their different demographic trees. And these countries were chosen to have a good balance between high income and middle and low income countries. And also in terms of what we consider for vaccination, we assumed that vaccination takes like 3 steps, but we added a 4th step for the vaccination of children. So this follows the recommendation of the World Health Organization. So we start with the elderly population, and then we move to the young adults, and in the end we move to the children, and we have a stage for booster dose. So the problem is in which time we need to move from this stage to this stage. So this is what we try to optimize for this specific problem. So we had to make some efficacy assumptions. So here we have considered a relatively high efficacy for the vaccine, but as a sensitivity analysis, we also consider like situations where the vaccines have a lower efficacy, like the ones which are commonly used for the low and middle income countries. So this is what the vaccination coverage for different ages looks like. This is for the booster shots, and this is for the primary vaccination. We also made some assumptions, but at this time, Omicron was not did not emerge yet, so we had only Delta. So we had to make some assumptions regarding the incubation period of Delta, the hospitalization ratio, and the escape to vaccine protection, and also some other assumptions. And we also consider the scenarios that you know. So scenario 1 is slow immune waning, scenario 2 is moderate immune waning, and scenario 3 is fast immune waning. And so this is the results that were obtained. So what we did is that we made a simulation under the same conditions, so without fitting to the data, and then we tried to see what happens when vaccination starts. And it seems that for the averted cases, so this is the number of cases that could have died but did not die, then it seems that for the low and middle income countries, it's better to switch to boosters a little bit early than for the for the high income countries. So it's like 90 days for the low and middle income countries, so Brazil, Peru, and South Africa, and for France, the United States, and Korea, then it's better to wait a little bit and then switch to boosters. And the reason for that is that these countries, they have a higher population of high risk group, and so it might take a while before you need to vaccinate them again. So yes, and this is just for the other for the immunity. So this is for the slow immunity. This is for the moderate immunity. We have the same conclusions, but the timings are a little bit different. And this is for the fast immunity, where we have both all countries basically need to switch to boosters sooner. And this makes sense because for fast immunity, of course, this high risk individuals, they lose immunity very fast. And so you need to introduce immunity like 3 months after stage 4. So we show that there is a timing window for the introduction of booster dose, and in some situation, then for low and middle income countries, it's better to introduce the booster shots sooner than the high income countries. And we had this meeting with this group, the World Health Organization SAGE group, and it seems to agree with the recommendations that they had, and that they found using different models like the one they are using at the Imperial College of London and so on. But we also had to do some sort of sensitivity analysis, like, for example, the effects of vaccine hesitancy. So what happens if more individuals do not agree to take the vaccines? And it seems that this does not change the like the dynamics or the timing, but it only changes the number of averted deaths during vaccination. As I said at the beginning, we also wanted to look at what happened when we consider vaccines with a reduced efficacy. So we had these two scenarios: A, where we have this protection that we had at the beginning, and B, where we have lower protection. And as you can see here, this does change the dynamics of the model. So when we use vaccines with lower efficacy, we need to introduce boosters sooner, even for high-income countries, as you can see here for in this low three curves. All right, so that was the second part. So where we have used I don't know how much time do I have.
Well, in fact, our official time is over. If you need several minutes more, we can continue now. Otherwise, if you need more, we can just do another seminar for further results. So it's up to you.
Yeah, I would try to just conclude in like 2 or 3 minutes. So we had this request from the Center of Disease Control, and they wanted to know what would be the impact of Omicron on the United States. And so at that time, there was a lot of uncertainty whether this high growth rate of Omicron is attributed to transmissibility or immune escape. And so we come up with this other version of the model where we have 3 immunities, one from vaccination, one from Omicron, and one from Delta. We have made some modification to take into account the Omicron growth function and how it modifies the parameters. And we made some scenario projections. But what I want to say, so this were the projections that we made on January 6th before the emergence of Omicron, these projections were very accurate. So one of them was because it's scenario projections, so we had 8 scenarios, and one of them really could tell the timing and magnitude of the peak of reported cases, hospital admissions, and deaths. And this is very interesting because even the other models couldn't tell this for sure. And so now currently we are using the model for all type of studies, like what's going to happen after the wave in the future, and whether immunity can withstand the emergence of new variants. And what we found is that if there is a new variant that is 50% more transmissible than Omicron that's going to emerge on May 1st, then the speed of immune waning will play a major role. Because if, for example, you can see here in this green curve, so this is what happens if immunity wanes very fast. Then if there is a new variant, we will see a new surge that is very high. But if immunity wanes slowly, like in these 2 curves here, then it will be able to withstand the emergence of this new variant. And the difference in terms of hospital admissions is very huge. So it's like more than 5 times difference between immunity between like the peak number of hospital admissions when immunity wanes very fast and when it does not. So this shows us that the studies on immunity gained through natural infection with Omicron is very important, and at this stage, but the recent trends suggest that we are heading toward the scenarios where immunity is quite robust and long-lasting. And as I said, we are still continuing this work and evaluating what could happen when there are behavioral change, and also we have this subvariant BA2, I think also here in the US they will start vaccinating the kids between 0 and 5 years old. So as general conclusion, what I want to say is that so as you can see, we have developed this new framework that integrates population immunity into COVID compartmental classical models, and this seemed to really solve a lot of problems in terms of complexity and high dimensionality. This means that it makes the results interpretable, and this can be used to have to give us an insight on the interplay between variants, immunity, and vaccination. And currently we are relying on this framework to inform the public health officials at the local levels and also the national levels. So we have published already 3 reports, 2 of them for the Omicron emergence in the United States that were devoted to the CDC and the White House. And one of them was for the local authorities here in Austin and Texas. So that's everything. Thank you for your attention.
Well, thank you, Anass, for this really very interesting and very intense presentation. You kept our attention really all this time. So we have some questions. Mali, please, you can ask your question.
Yeah. Anass, thank you very much for nice and excellent work. I just wanted to ask one question that might be a stupid question also. That in your work, if I understood correctly, that it is assumed that vaccine or booster dose that would be effective for new emerging strain. But if it does not happen, that if this existing vaccine does not remain effective for upcoming new strains, then what will be the strategy?
Yes, thank you very much for this excellent question. And yes, you are completely right. There is always possibility for the emergence of variants that escape to Omicron immunity and even to the vaccines, because Omicron already escapes to the vaccines. And as that time progresses, then there will be few immunity gained from the vaccines, or it will be less effective. So we are currently studying this question. So we are participating in a consortium here in the United States called the Scenario Modeling Hub. So it's like a group of teams working on the modeling of infectious diseases. And we are organizing rounds each month where we consider different scenarios where, for example, we change the speed of immune waning and the rollout of booster dose, or, for example, the characteristics of Omicron, depending on the tasks that we want to study. And we are going to study this question for the next round. So you are absolutely right. I think there are good reasons to think about the emergence of variants that escape immunity and what's going to happen. And I think in this case, we still need to rely on some level of immunity, because if we didn't have this immunity and had Omicron, then probably the situation will be more catastrophic. But since we had this immunity, it seemed to be working against hospitalization and death. So we had this high level of infection, but only few of them require hospitalization and death. And this will probably also translate to the next variant. So probably if there is an immune escaping variant, then if we have high level of population immunity, then there will be still a few people that will go to hospital or die. I think there are different strategies that can be used depending on the context and the resources that we have. But it's always good to have high level of immunity.
Yeah, and one more quick question, that also you have assumed that next dose administration, that means booster dose, depending upon the efficacy of the vaccine. But do you have any idea about the literature, that is level of antibodies within individuals after giving the vaccines, or for different age group, or maybe some country specific data?
Yes, so if I understand your question correctly, so you are asking about the level of antibodies about after getting vaccinated. So what is the level of antibodies?
Yes.
Yes, I think there are some reviews that give some estimates. But I think there are a lot of variability. So there are people who do not generate any antibodies, and there are people who generate a lot of antibodies. And this is why we have some level of breakthrough infections. But in terms of modeling, so the number of antibodies need also to be correlated with the efficacy of antibodies, or the capacity of neutralization of these antibodies. So, for example, we know that for Omicron, you need to have about 2.4 level of antibodies higher to prevent reinfection with Omicron than Delta. So yeah, so we have some data like that, but I think that there should be some more clinical studies showing the level of antibodies. Yes.
Okay. Thank you very much. Maybe I'll write some email to you for further queries and discussion. Okay. Thank you. Thank you very much for your nice talk.
Yeah, please do. Thank you very much for the questions. Thank you.
Yes, please. Other questions in the audience? Well, in fact, I have, well, the whole list of questions or remarks. Certainly, we cannot discuss all of them now, so maybe I'll just keep only one or two of them. There are really interesting questions to discuss. In particular, you mentioned in the beginning of your talk about the relation of viral load and symptoms. You know, you showed these curves, like dependence between the two, viral load, hospitalization, things like that. I think it's really a very interesting question and maybe not sufficiently discussed, or maybe it is, I don't know. Because as far as I understand, what is viral load? Viral load is, roughly speaking, how many virus particles are there in the organism. It's a number.
Yeah.
It's not symptoms. It's just a number of viruses. Symptoms is tissue damage.
Yeah.
And this is not exactly the same. We can have more viruses and less tissue damage, or vice versa. Yes. So, and in fact, it's exactly the question we work on now. So, to shortly summarize, is that there are two different things. There is viral load, but there is infection spreading in the tissue. Okay.
Yes.
The speed of infection spreading, and mathematically speaking, infection spreads as a reaction diffusion wave. Yeah. So we have the wave of the speed. Sorry, what I say? The speed of the wave. The speed of the wave. So simplifying, symptoms correlate with the speed of the wave, but not with viral load. These are two different things. And this is a very interesting question. So if you are, well, I will not develop on that now more. We don't have enough time. But if you are interested, we can return to this question later.
Yes, I will be very happy to discuss later. And I completely agree. I think it would be probably more interesting to even consider the number of infected cells, assuming that some of them will be damaged.
Exactly.
But yeah, and but here, for example, in this works that I gave for influenza, they use this area under the curve because it's just an indicator, like a clinical indicator, and they found some correlation.
Exactly. Absolutely. So again, if you are interested, we can discuss it later. But what I just, again, very shortly, if we take one given virus variant, for example, Delta or whatever, for the same variant, there is a correlation between viral load and symptoms for the same variant. Meaning there is a correlation between viral load and the speed of propagation. Because in fact, modeling all that, we have analytical formula for all the things, for the viral load and for the speed of propagation, and we see how they correlate. So for the same variant, they do correlate. And we have more viral load, more symptoms, more hospitalization. But if you take two different variants, it can be, like you take Omicron and Delta. Omicron has larger viral load in the upper respiratory tract.
Yes.
And because of that, it has larger infectivity.
Yes.
But the speed of spreading, this is completely different. It is the speed of infection spreading, it depends on cell types. And we do this modeling and we see that the speed of spreading for Omicron is larger in the upper respiratory tract, but is smaller in the lungs.
Yes.
This is why symptoms for Omicron are weaker. Large effectivity, but weaker symptoms. It is because viral load and spreading speed, these are related but different things.
Yes, yes.
This is shortly speaking. Okay, but it's really interesting. And of course, yeah, many other things you mentioned. I like very much this model with where you introduce this population immunity, this variable M, and also like this M, which like grows in the beginning with some overshoot.
Yes, yes.
And then converges to the stationary state. It's also a very good point. Okay, not to make our discussion too long, I have the last question, which is half scientific, half philosophical. So as far as I understand, one of the results you presented shows that we cannot eradicate completely this infection. And well, even simple models show that, yes, indeed, it is like that. If we assume that susceptible, then recovered become again susceptible, then for the basic reproduction number larger than 1, we have this endemic equilibrium, positive endemic equilibrium, to which we converge for a long time. This is, in simple words, the dynamic of the disease. At the same time, if you introduce vaccination in the population, then it depends on the level of this vaccination. If the level of vaccination is high, we can reduce this endemic equilibrium to the number less than 1. So there is a difference between mathematics and like applications of mathematics. In mathematics, numbers less than 1 exist, no problem with that. But we count people if there are like, we obtain the results, the endemic equilibrium, the number of infected people is less than 1, then the epidemic is finished. So when you say that we cannot eradicate it, do you take this into account?
Yeah, if I understand correctly, so you are saying that if the vaccination intensity is very high, then it's possible to have the basic reproduction embryo.
No, sorry. What I say is that if we have like then we will eradicate completely infection because the number of infected people will become less than 1.
I see. Yes, I understand. Yeah, I think the problem here is that when you have a continuous model, then even if you have quote unquote 0.1.
Right. Yes. This is why in this continuous model you put very simple additional condition. When the number of infected people is less than 1, you stop simulations and you say it is 0. In fact.
Yes.
You see what I mean?
Yeah, I completely agree, but I think in reality there is this super spreading. So we know that like people, there is like this short period of time, like 1 day before symptoms, where people have very high viral load and they can spread the virus everywhere. And so we have this super spreading event, and yeah, so I think the reality is much more complex than.
Of course, it is complex, but I don't think this super spreaders. It's a little bit different. Just the last argument to convince you to make you more to make all of us more optimistic, because we need a hope. Because from general point of view, there are infections which are completely eradicated. You agree with that?
Yes.
There are examples of viral infections which are completely eradicated.
Yes. But
so we, of course, if we take like influenza, for example, why we have all the time this emergent every year epidemics? Because these are new virus strains.
Yes.
We never have exactly the same strain. It's new strains which produces new epidemics. Of course, if for this coronavirus, if all the time new and new strains will appear and will escape immunity, then of course we will never eradicate it. This is clear. But if it is possible to develop a vaccine which is efficient for the whole group of variants, and in fact, in literature there are some signs, there are some studies now that there is, maybe it will work, nobody knows. It's very preliminary, it's on mice, on animal studies. Maybe there are some vaccines which can be like act in a more general way. So to summarize, let's hope for the best. Okay, Anass, thank you very much again for this beautiful presentation, very rich and very well done. There are no other questions in the audience, so we stop on that our seminars, and we'll continue next time with our next speaker. So everybody is very welcome. And I think Anastasia, do you have anything to add? No. Okay, then thanks a lot to everybody. We stop on now. Bye.
Thank you very much. Bye.
Thank you. Bye-bye.